Okay, we're going to start Lab 209. Here's the lab sheet. It's at the bottom of the 208 lab sheet, so just keep scrolling down. Here's the website to click to get the application that you need. It should run with the same launcher that you launched Lab 208 with. And let's start with the first page. Just like the last lab, question 1 is going to line up with page 1. We're going to take a look at some nucleotides and talk about DNA. So here's what it should look like when you launch, and we'll go to page 1. So you can read all about how you can pause the recording and read all about how DNA is built from nucleotide monomers. Now a monomer is a link in a chain. In the case of DNA, it would be one of these. Connected to one of these, connected to one of these. These are all monomers. They're part of a polymer. Poly means many, so it means many mers. It means many monomers. You can see that my model DNA here has lots of individual units all connected together that make a polymer. The parts of the polymer of DNA are called nucleotides. Let's look at the 4 nucleotides here. There's just 4 of them, just like the 4 colors. There's adenine, thymine, guanine, and cytosine. What's different, you can see, is the base here. This part of all of them, all 4 of these letters here, are the same. It's this part up here. Here's a DNA fragment. It's basically half of the twisted ladder. We're going to explore the structure by clicking and dragging the mouse. You can highlight the nucleotides if you want to, and highlight the monomers in different colors, and you'll notice those turn different colors too. So T is yellow and A is blue. To discover the sequence of the nucleotides, which is question 1, you can say 1 is the blue one, which is of course C. Number 2 is C. Number 3 is A. So now you know how to answer question number 1. What's in a nucleotide? Well, let's look at what's in adenine first. Remember I said all this stuff is the same. Let's check out, this is the phosphate, this is the sugar that makes up the nucleotide. There are 3 parts: the phosphate, the sugar, and the base. Now as we click through and look at different nucleotides, the base changes. So there's 2 rings here, 1 ring here, 2 rings here, but there's 2 functional groups there, and 1 ring here. The elements for all of them are the same. They're all carbon, oxygen, and they don't show the hydrogen, but they do show the nitrogen. And the phosphorus. That's the orange one. That's kind of new for us. Which components are the same in the DNA? Well, I'll click through one more time. The green and the purple stay the same. So you can look and see what's green and purple here. Which components serve to link the DNA nucleotide monomers together into a polymer? Well, these bases connect to each other in the middle of the DNA. These guys link to each other. They're the sides of the DNA, like the sides of the ladder. This represents the sugar and the phosphate here. All right, let's go to question 2. You can read all about this and pause it. We're going to look at hydrogen bonds between the bases. So here in my model, you can see that the bases come together in the middle, like this red and yellow one, and they stick together. The plastic is linked here, and that represents a hydrogen bond. We're going to look at, this is kind of a very big overview of DNA. We're going to look at a more molecular view. We're going to try to match up the bases here, and guess what? two bases, you can see that the pairing is always the same. Here, it's always blue and green and red and yellow. Doesn't matter if red's on this side or if yellow's on this side, but yellow and red always go together and green and blue always go together. So let's see what those colors represent. Let's try to make some hydrogen bonds here. That's not going to work. We're going to see dotted lines develop. So that works okay. Let's see if that works better over here. Nope, we're trying to make the maximum number of hydrogen bonds. So A and T go together, they make the maximum number of hydrogen bonds here. How about A and C? Nope. Nope. Oh, that looks like a great fit with 3 of them. T looks like it fits well with A, and that makes sense because A and T go over here, and it makes sense that G would go with C like this. So now that we know those pairs are G and C and A and T. What's the greatest total number of hydrogen bonds you can form in the model? Well, let's look. Just count the dotted lines. So 1, 2, and I'll let you count the rest. Using what you've learned about fitting the bases as opposite pairs, predict which bases you'll find with each of the 4 bases in the double DNA helix. We've already answered that one. So for question 3, you can read all about how 2 DNA strands wrap around each other to form a double helix, like a twisted ladder. And here we're going to basically mess around with the different controls until we find something that lets us see those hydrogen bonds in the middle of the molecule. So here's our DNA molecule. We can kind of twirl it around here, and we can't see hydrogen bonds yet because we're in space filling. Let's go to sticks. Maybe they don't really show up in sticks. Let's go to ball and stick. You might be able to see a little bit. Let's go to ladder. Oh, it's because we don't have hydrogen bonds. So you can see the little dotted lines in the middle, like right there and right there. Those are the hydrogen bonds. Remember, hydrogen bonds are between molecules, not between atoms. So they're different than regular bonds within the molecule. They're between molecules, so we show them that as dotted lines. If we show chains here, those hydrogen bonds show up a little bit better. You can see how the yellow side, or one side of the DNA, it's not really yellow in real life, but we use the model here to be able to see it better, is bonded at the base with the other side of the DNA. So there's 2 sides of these ladders, just like our model here. The 2 sides of the ladders would be the yellow and the purple, and they twist up, and in the middle here, the 2 bases come together and form hydrogen bonds. And this is our best view here, so you can take a screenshot. Okay, time to switch gears here a little bit. We're going to go over from DNA, this molecule that's always the same shape and only has 4 monomers, to a molecule that's a much more complicated shape, depending on its function, has lots of cross links, and has 20 different monomers it's made out of. There's lots of crazy things. 20 amino acids with lots of different shapes and different, they're not really different colors, but we represent them as different colors here to show how different proteins are from DNA. They're both polymers, they're both made of the same repeating units, but they have different shapes and different functions. This is pretty much right out of the state standards. Proteins do much of the work in cells, serving as enzymes, that's catalysts to make reactions happen. They serve as structural supports like collagen, it supports your nose. Transporters, they get things around the cell and your body. They tell your cells what to do as messengers. So this is really important. Proteins are really important. Let's watch a movie that shows you how important they are. So you can see all the different stuff that goes into making a protein. They're molecular chains made out of amino acids. And they're just coloring the chain, not to show the different amino acids, but to show kind of, so you can see the different parts. That's myoglobin, that's one of the questions. It carries oxygen in muscle cells. So for 4A, heads up. We rely on myoglobin. This is another cool piece of protein that can grab onto a piece of DNA. We call it protein tongs. That's 4B, heads up. So this is the DNA, this is the protein. Very different. You can see the different structures here. The protein is forming the X. It can make lots of different shapes. The DNA is always this double helix shape, this twisted ladder. Here's a really cool protein called porin that creates like a pipe in the side of a cell membrane, or a channel, as you will, to let things in and out. It's also a molecular chain made out of amino acids. DNA is made out of nucleotides, 4 nucleotides, and a protein is made out of 20 different amino acids. And the order of those amino acids determines the shape of the protein. We can try to follow it all the way to the end. You can kind of see how you can kind of follow the blue, and then you can follow the light blue, and then the turquoise, and then it starts to kind of turn green here, and you go into this lighter green, and it zooms this way, then it turns kind of yellow, and we'll turn it around here so you can see the yellow better, into the orange, into the red, and there's the end. Kind of cool, huh? It's really just one long string, but it makes this channel, or this kind of pipe shape, cylindrical shape, in the side of a bacteria. So proteins have shapes according to their jobs. Which one do you think forms a cable? I'll give you a hint. It's the one that provides a lot of structural stability in your body. Your ears, your nose, your skin has a lot of this cable-y protein that's very supportive. Which one of these three do you think can puncture the side of a bacteria? Which one has a shape that looks like a function of something that can poke a hole? And which one looks like a channel or a pipe? So which of these, 1, 2, or 3, can puncture a cell wall? Which protein forms a cable? And which protein becomes a pore in a membrane? A pore is another word for like a pipe or a hole. So now we're on question 5 here, because we're on page 5. You can pause the video and read all about how DNA has 4 monomers, and proteins are made from 20 different amino acids. Not nucleotides, proteins are made from amino acids. Let's play this movie and find out more about the polymer chains that are called proteins. We're going to look at porin again. And they just make it rainbow color there so you can kind of follow the chain. We'll look at a small piece. Each part, each monomer is called an amino acid. So proteins are made out of amino acids, DNA is made out of nucleotides. The part that branches out from the amino acid is called the R group or side chain. It's like the base in nucleotides. It's what makes it different. Amino acids are all pretty much the same, have a part that's the same and a part that's different. And if you want to think of me, your science teacher, or one of the science teachers in the department is a little weird, kind of different, that's Miss R. And the R group, of course, is a little different. So amino acids have those different personalities, and that's because of their R group. That's the different part. An important part of that R group, or that different part of the amino acid, is its polarity. So let's check out the polarity of a couple amino acids here. So let's look at the surface charge. And we can look at the whole amino acid. You can see that the charges are really only on the side chains. And let's look under the surface charge here and see there's 2 nitrogens here. And if we put the surface charge on again, we can see that those nitrogens create a positive surface charge. Let's turn it off again. Neutral surface charge right here, carbon and hydrogen. Let's look again. So this is pretty neutral. This is valine. Let's see what's under this one. It's positive blue here and negative red here. What do you think is going to be negative? What is usually negative? It steals the electrons. Yeah. Red's oxygen, and oxygen's an electron hog and tends to be negative. So now we know about those R groups, those different parts of the amino acids. And so what makes it polar? Well, there are different atoms here that have different polarities, and so they cause the side groups to be different polarities. So for question 5C, you're asked to figure out the charges of different amino acids. There's 20 amino acids listed here, and the charges here are non-polar in red, and blue is polar. Positively charged, that's of course going to be polar, and negatively charged, that's of course going to be polar. So if it's red, green, or blue, it's polar. If it's kind of this pink color, it's non-polar. So I'm going to scroll up here so that you can see all the different amino acids. So there's 20 different amino acids, and you can count how many are non-polar here, that pink. And the rest of the colors, whether they're positive or negative or just marked as polar, which probably means they're both positive and negative, they're this blue color. That counts as polar. This counts as polar. This counts as polar. And this is nonpolar. We can simplify it just like this, going pink and blue, which is nonpolar and polar, which is hydrophilic. The polar parts, the polar amino acids are going to love hanging out with water, and the hydrophobic parts are going to want to hang out with the other hydrophobic parts of the protein. So here's another look. You can pause and count if you'd like. So now we're going to think about how the watery cytoplasm of the cell, the inside of the cell, how does that affect how proteins kind of crumple up, kind of form themselves? into different shapes to have their different functions. And that depends on the R groups here. We can see this is valine. We looked at it in the last screen. It's a nonpolar amino acid. Let's run this and see what happens. Do you see the valine really interacting with the water? Is it making any hydrogen bonds? These little dotted lines among the water are the hydrogen bonds. I'll turn them off so you can kind of see the difference here. We can turn off charges too so you can see them better. I don't really see that valine making any hydrogen bonds with the water, and it's kind of sticking with itself. So in water, the hydrophobic parts of the protein, the amino acids that don't make hydrogen bonds with water, like this one here, like valine, something's going to happen to them. Let's watch what happens. I'm going to run. The pink parts of this protein are hydrophobic, like valine that we saw above. So they get pushed to the middle, and the more polar amino acids, these blue ones, hang out on the outside. Let's randomize this amino acid and see what happens to different ones. You can see that you get different shapes as you get different combinations of amino acids in different orders. We'll give this one a little time to organize itself. So this protein is going to have a bit of a different shape because it has a different order of amino acids. This one seems to have more hydrophobic. It's the order of the hydrophobic versus hydrophilic too. Here's kind of an idea of, here's a good model. Here's lots of different amino acids in a chain. We'll call this an R group. These things that are kind of hanging off, we'll call the R groups, or especially large R groups. And these R groups like to stick together too. And that helps make the protein chain as well. That gives the protein its shape. This one might be like porin and making a channel. This one might be more like hemoglobin, which is a very complex protein in your blood that carries oxygen. So if there are many nonpolar or pink amino acids, hydrophobic amino acids, where do they end up? Well, let's pick another one and see what happens. They tend not to end up at the edges. So explain how the interactions between water molecules cause the hydrophobic amino acids to fold into the center of the protein. Well, basically, the polar or hydrophilic or water-loving amino acids want to hang out with water and make some hydrogen bonds. Everybody else doesn't. They'd rather hang out with each other. The hydrophobic amino acids like to hang out with other hydrophobic amino acids. So there's your thought for question 6. All right, at this point you should have enough information to answer question 7. We're on the home stretch here. This is the last question, but I'll give you some hints. What causes the 2 strands of DNA in a double helix to attract and wrap around each other? So remember what we said, these 2 bases joining together with this little clip here, that represents hydrogen bonds. And the hydrogen bonds don't happen between the sugars and the phosphates. They happen in the middle. Which part of the DNA nucleotide carries the genetic information? Well, it's the order of the bases. These things in the middle, these things that are different from each nucleotide. The side chain of an amino acid Remember, it's the Miss R. It's kind of something that's different. I'm a little different, so when you think of the R group, think of something a little different. And think of me. A protein chain will fold depending on its side groups. Remember, these little things can bond to each other. Or here's another representation. This can serve as a representation of a disulfide bond, which we'll study later. So a protein chain folds into a shape that's related to what it does. It's a structure-function relationship. So, big question here, how are the structures of DNA and proteins similar to each other, and also make sure they're different? DNA is made out of nucleotides. There are only 4 nucleotides. It always has the same shape. DNA is always a double helix. No matter whether you're a frog or a pig or a human or a bacteria. Well, maybe not bacteria, but if you're any mammal, your dog is DNA that's this shape. Proteins are all kinds of different shapes. Some are kind of lumpy, some make channels, some kind of just crumple up into a ball here and are kind of just a big mess. This is a good model for a protein because it's got a bunch of different colored beads here. So the structures are different, the shapes are different. They're both polymers. They're made out of repeated units. There are 20 repeated units in proteins, only 4 in DNA, 4 nucleotides. The protein on the right here is folded up. Basically, you need to explain what's in the middle and why it's in the middle, what's on the outside, and why it's on the outside. All right, you've got everything you need to know to finish Lab 209. Thanks so much. This is Miss R signing off for Lab 209 in biology.